Welcome to OA Week. We'll give you a few more minutes to sign in, wait for a few more participants, and we'll get started in just a minute. Okay.
Yeah, Iris is the participant. Yeah, she's.
Yeah, I think we just let her in.
This is Iris, yes.
Some slight technical problems. We'll be right with you. Amazing. Okay. All right. So welcome, Iris. It's good to have all of our moderators and co-presenters present. Excellent. Welcome everybody to OA Week 2024. You're now in the Mediterranean Hub, Stefan. And just for your attention, this session is being recorded. So I would just quickly like to introduce you to OA Week. My name is Katarina Shaw. I'm with the Intergovernmental Oceanographic Commission of UNESCO, and I'm also part of the GOA-ON Secretariat, which is why it's my pleasure to be able to welcome you here. OA Week is presented by 4 sponsoring organizations. First, we have GOA-ON, obviously, the Global Ocean Acidification Observing Network. Secondly, we have the IAEA OAICC, which is the International Atomic Energy Agency Ocean Acidification International Coordination Center. We also are sponsored by NOAA, the United States National Oceanic and Atmospheric Administration, and last but not least, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organizations. So GOA-ON was established in 2012 with just a handful of members, and since then has grown enormously. We now have over 1,200 members from around 114 countries, and we also now have 10 regional hubs, one of which we're about to hear more about. And we have a membership sign up. So if you're not a member of GOA-ON yet, we invite you to come to our webpage and sign up. We have started this OA Week in 2020 and returned in 2021 when events and conferences were postponed or cancelled due to the COVID pandemic. And following the successful in-person symposium on the ocean and high CO2 world in 2022 in Lima, Peru, and another successful OA Week in 2023, the community asked us to repeat the exercise, and so we're happy to be back. We have yet again another amazing lineup this week, so we hope you're going to check out the webpage and the registration links. There's more to come. So a little bit of housekeeping for you all. During the presentation, all participants are in listen-only mode. You're welcome to use the Q&A box to type in your questions. And you can also raise your hand by using the raise your hand button, and then we can call on you following a presentation. And with that, I would like to hand over to today's moderators. We have Iris Hendriks from the Mediterranean Institute for Advanced Studies in Spain, and Abed Rahman Hassoun from the Helmholtz Center for Ocean Research, GOMA in Germany, and the National Council for Scientific Research of the Lebanon. Over to you.
Thank you so much, Katerina. Thank you everyone for being here. And yeah, it's such a pleasure actually to see this number of audience and to share actually the latest updates of our science and our colleagues here during this session. So I'm not gonna talk so much about the OAMed Hub. Most of you know our activities and updates that we share with you on our social media accounts and the newsletter. I will just ask my co-chair Iris if she wants to start with anything before introducing the first speaker.
No, I think you got it all covered, Ahmed.
Perfect. So it's time. Actually, I'm super honored to be introducing today 5 actually female scientists here and for our OEMED Hub session. And I'm going to start with Maria Cristina Gambi. Maria Cristina Gambi is actually a researcher affiliated to OGS, the National Institute of Oceanography and Applied Geophysics in Italy, but also to ISPRA Roma as well. So Maria Cristina graduated, basically she is a biological oceanographer, and she started super early with her expertise in oceanography in general since 1983. And she worked on many topics related to systematics, ecology, and reproductive evolutionary biology of polychaetes, marine worms. Maria actually wanted to basically initially to talk about polychaetes and potential effects on polychaetes, but now we have changed this, but we hope that in the future, Maria can also deliver for us this originally a planned actually presentation, because I'm sure that it's going to be interesting for many of us here. So she worked as an editor for many journals, and she is a member of the Italian Society of Marine Biology, and she co-authored many papers. I'm sure that those who are working in the Mediterranean Sea already know Maria Gambi. So you are more than welcome, Maria, and the floor is yours.
Thank you very much. Thank you very much for the presentation. Can I share my presentation? Can you see now the presentation?
You're not in presenter mode yet, Maria.
No? I'll try again. Did you see now?
We can see your slides.
Maybe if you just disconnect and then reconnect again, the way that you've done it before, that might work.
Excuse me. Yeah. Yes. Yeah. Then I don't know how to connect again. Now I see the presentation, but I don't see the command to share the screen. Sorry.
No pressure at all. So you only have to share, just to press on the green arrow. Yeah, perfect.
So
and now try to share your screen.
I share the screen now. Did you see the presentation?
The presentation mode? It doesn't work?
Yeah, I press the button. I see the screen full, but you don't see it. I don't know.
No worries. We are able to see it this way. Maybe you can proceed with it this way, because we are able.
I'm sorry. I can try again. Now, no. Sorry. You see the slides. You don't see the presentation, the full screen. Sorry.
No worries at all. Don't worry at all. So you can reshare your screen, Maria, because it's not shared.
It's not shared. Sorry. So I get confused because I don't see the Zoom anymore now.
It's okay. We all have lots of tabs.
Okay, I can share the schermo, maybe. Okay. So I try this way. You see now?
Yes.
Sorry. Okay, sorry about I'm
Perfect.
a little.
The floor is yours. You can start.
Yeah, so sorry about that technical problem. Thank you for the presentation, and I am very pleased and honored to have the opportunity of this talk. I will shortly do a synthesis on the CO2 vents as natural laboratory to study the effect of ocean acidification, giving you an overview of the system we are currently studying in Italy. As you know, the CO2 vent of volcanic origin, they show a continuous flux, mainly of CO2, but not only. We will see there is also other gas, and which is the CO2 is causing an acidification of the local surrounding water. And since almost approximately 20 years ago, since 2006, I will explain you why this shallow two vents system have been intensively studied as ecological analogs and natural laboratories to evaluate the effect of ocean acidification on the local biota. Why since 2006? Because the first studies started in that year to address this topic and was done at the Castello Aragonese events in the East Island, where I worked for almost 40 years, because I was working previously at the Stazione Zoologica of Naples. And this is the famous paper of Paul Spencer et al., which appeared in Nature in 2008. And from that point, there was an increasing interest on this system. And the last review, which concentrate on the geochemical conditions and of other coastal vent system conditions which are suitable to study the effect of ocean acidification, we have now 55 potential new systems. Among these systems, however, the vent studied from a biological point of view are less, are 26. Here in this review paper of 2018 have been listed 20, but recently other 4 vent systems in Greece and in Italy also, and in the Canary Island and other places have been described and have been studied, I repeat, from a biological and not geochemistry point of view. So these are relatively a few, if you think respect to deep hydrothermal vents, which are much more studied, or cold seeps, which are more studied and more numerous, simply because studies have started relatively recent on this sense. However, respect to this, we have already various review, and which include sometimes not only the CO2 vents like these two, but generally hydrothermal vents, but there are review concentrated, for instance, this is on boreal system, Rastrig. This is a worldwide review, which an update more recently. And this is a synthesis, Ayupa and Tal, which I showed you before, is a synthesis on geochemistry and the synthesis on the volcano vents. This is a review on the Castello vents, and these are other review on the importance of hydrothermal vents for biotechnology and for conservation. And I participate as an attender to the ICONA meeting, which was a previous meeting today. And I would like just to show that there is also this ICONA, this network of CO2 natural analog network. So let's focus now on the Italian situation. Italy is in the middle of the Mediterranean. In the Mediterranean, we have all possible hydrothermal vents described, from cold seeps to deep hydrothermal vent. And Italy in the middle is full of this system, but this is the atlas made by the ISPRA, by my institute, by the geological section of my institute. But the CO2 vents mainly studied are concentrated here in the Campania region in the Gulf of Naples and in the North Sicily in the Aeolian archipelago. This is the situation in the Gulf of Naples. We have participated to this atlas describing 7 different CO2 vent systems. And in the Åland Island, the events studied are mainly concentrated at the Panarea Island and the Vulcano Island. And but let's start from the site where all these studies started. That was the Castello Aragonese. Is a volcanic islet. Is the remain of a volcano connected with the artificial road and bridge to the main island. And from the islet to the main island, there is a geological fault. From this fault, we have volcanic emission of CO2, mainly CO2, 95% CO2. There is no sulfur, no toxic gas. Temperature and salinity are the same of ambient condition. And since we have different intensity of bubbling, we have a gradient from high intensity bubbling, both in the south, in the north, in this red area. And then we have a decreasing bubbling in intermediate zone with intermediate pH and the control zone outside of the vents. So here you see the gradient. Here you see at the image picture above of the very intense area. This is the intermediate area. And you see there is a gradient, and we established in the past 3 main stations, both on the north, on the south, with different degree of acidification. So we have a gradient. Just to give you an idea of the pH, here are the 3 zones, the control in blue, the intermediate in yellow, the acidified in red. And you see, first of all, you see that there is a strong fluctuation of the pH, both in the south and in the north. And this fluctuation are due because these are open systems. So we have tide, and we have especially here is Ischia, because there is a huge coverage of macrophytes and including Posidonia, dense Posidonia beds. These fluctuations are probably due to respiration at night and production of oxygen during the day. So these are the main macrophytes. The vents of the Castello are dominated macrophytes by dense Posidonia, but also brown algae, especially Dictyota and Alopteris and Schistoceira, and also some Caulerpa, including some alien Caulerpa cylindracea alien species. And these are the main invertebrates which are able to thrive under ocean acidification, vertebrate and fish. Here are which these organisms, these worms and small crustacean, very mobile, and fish are the object of various studies, and some of them have been used as model species to study the effect of ocean acidification at various levels, mainly ecophysiological, but also life history, and recently also at genetic and genomic level. We have synthesized the information at the Castello that now are 20 years, almost 20 years under study. We have synthesized 82 papers which somehow have a checklist or biodiversity data, including the description of 4 new taxa that have been described in the vents, and 8 alien taxa that have been found in the vents. And if you see, the majority of this biodiversity is concentrated 87% in the ambient zone, while going to intermediate zone with pH, or mean pH of this level, which is the pH projected for the end of the century by geochemical scenario, future scenario, we lost 49%, almost half of the species disappear. And if we move to the very acidified area where pH is generally below 7.8, we have a reduction, a loss of 67% of the biodiversity. Mainly, this is due to the loss of calcifying organisms, especially if we are sessile organisms. But in Ischia, we are quite lucky because we have other CO2 vent system, other 4 beside the Castello, And this system are the Vullatura, Chiane del Lume, Grotta del Mago, which is a cave, the Wizard Cave, and the Madonnina, which is a deep coralligenous bank. So we have a depth range of 2 in the Ischia vents to 48-meter depth, the coralligenous outcrop, which is representative of the main Mediterranean habitat and very common organisms. So we have a large array of organisms where we can work with, and we have represented shallow and deep rocky reef, shallow and deep Posidonia meadow, semi-dark cave, and coralligenous outcrops, including
We only
also
have 1 minute, Maria.
soft bottom. So
Thank
these
you.
are the 1 minute. Sorry. These are some of the picture of our vents, and this again, I show you the Vullatura variability also in the Vullatura system. Just move short quickly to the Panarea Island, which is a volcanic arc, and there are still 4 active volcanoes: Stromboli, Vulcano, Lipari, and Panarea. Panarea in particular is the largest hydrothermal field in the Mediterranean Sea. There are 23 different systems. The most studied is the Bottaro crater, where there was an explosive degassing of CO2 near the Bottaro islet, where huge amount of CO2 and a crater was formed, which is this one, is a 20-meter diameter and 10-meter, 12-meter depth, and the rim is 8-meter. And again, the bubbling is in the center of the crater, and then we have a decrease in the pH, and the Bottaro still we have a gradient of pH, although it's not so low as in Ischia, but we have still a gradient. And along this gradient, we have seen again dominated by macrophytes and associated small invertebrates, which are common. And at Panarea, we have other 2 systems under study now, which is the hot and cold point, and the Camp 7 system. Hot and cold are hot point where hot water, not only gas, but hot water, rich in sulfur. You see this green bacterial mat, which are interspersed with Posidonia meadow and with cold point, where there is no emission and there is no water coming from the bottom. And Camp 7 is very interesting because here is on a marine fault. The difference in depth is due to this fault, and along this fault there is huge amount of CO2 coming up on the water like geyser. So these are very spectacular system. And the volcano vents, to finish, in Levante Bay, they have been shown, is interesting because we have primary vent in the bay, which here is again, white is sulfur bacteria, and is so strong, the gas, that the water, like a fontaine, because it's very, very shallow, is coming up to the surface. And to study the effect of CO2, we have to move 300 meters from the primary vent and to have this still CO2 enriched site and fire control site where the majority of the sulfur is deposited. And again, you have the dominance of, of macrophytes, especially brown microalgae, Sargassum schistoceira. And I want to finish saying that although the natural CO2 vents cannot be considered precise analogs to study the problem at larger global scale, because have you seen they have a limited extension, limited depth, and a huge variability in space and time of the carbonate chemistry and of the pH. However, they integrate, they can give a useful insight because they can integrate effect on biogeochemical cycle over a long time. The Ischia vents have been estimated almost 2,000 years old. So to test model organisms and scale up laboratory and mesocom experiment, identify winner, the species that can survive, and loser, species that are selected, and show changes in measured species and ecological feature and interaction.
Thank you so much, Maria.
Along the gradient.
So much,
Thank you
Maria.
very much.
That was super interesting, but because of our very tight time, thank you so much for this very interesting contribution. Now our next speaker is Carlotta Dentico. Carlotta is actually doing her PhD between the University of Venice and also OGS. So we do have two colleagues here from the OGS. So she's specializing in marine sciences, and her research focuses on the dynamics of the carbonate system in the water column and air-sea carbon exchanges, mainly in the North Mediterranean Sea and the Adriatic Sea, actually. Through multiple oceanographic cruises, also she gained expertise in biogeochemical sampling, laboratory analysis, and the floor is yours, Carlotta.
Okay, thank you. I will share my presentation. Okay. Should be able to see. Oh, can you confirm that you can see my presentation?
It's fantastic. Fantastic. You can start.
Okay.
Thank you.
Perfect. So good afternoon, everybody. I will give you a synthesis of the main research activities that has been going on in the Adriatic Sea, in the Northern and the Southern Adriatic. And I'm giving this talk on the behalf of all the co-authors that you can see in the first slide. And at the end, I will just give some future perspectives and research questions that we would like to investigate and deepen more. So I will start giving a few slides of in introduction, describing the main carbonate parameters in the Mediterranean Sea. So starting from pH, when we compare the latest estimate of pH changes in different regions of the Mediterranean Sea, so in the Northern Adriatic, in the Gulf of Lion, or in the Levantine Basin, we can see that these decreases has been higher compared to the global ocean decrease that are highlighted by the red in the panel on the top left. And it is well known that the Mediterranean Sea accumulates higher anthropogenic CO2 compared to other oceanic regions, mainly due to specific physical oceanographic processes. Also, alkalinity, it has been observed that the Mediterranean Sea is characterized by a west-eastern gradient with lower alkalinity in the western part, where we see the influence of the Atlantic waters, and becoming higher in the eastern base, mainly characterized by higher temperature and salinity. There are, however, regional and also seasonal variability of the parameters, highlighted by this figure that I took from the paper of Cossarini et al. In 2015, showing the mean alkalinity at the surface, and these are model outputs. And similarly, when looking at the fluxes, we can immediately identify spatial and temporal variability in the different submasins. Reddish color identify basins that are more that acts more as a sink of CO2, while bluish color identify regions where on a annual scale there is more release of CO2. This is again a map taken from some model outputs, and these regional and also temporal differences can be due to meteorological forcing, biological activity, and also the characteristics of the different water masses that are present in the Mediterranean Sea. So it's a very dynamic and variable region. Here I will focus on the Adriatic Sea, and it's a basin that it's located in the northeastern part of the Mediterranean Sea, and it's a very important location for the production of dense water for the Eastern Mediterranean, the Adriatic deep water, which the main contributor is the Northern Adriatic dense water formed in the shallow area. There is indeed also a complex bathymetry with a very shallow area in the north and very deep area in the south, where the bathymetry reach more than 1,200 meters. It's a region characterized by the interaction of different water masses. There is the entrance of Levantine water or Atlantic water and Ionian water, which are strictly linked to the dynamics of the changes in the vorticity of the Northern Ionian Gyre, located here. It's a region characterized by a general cyclonic circulation, and there are two main currents in the eastern boundary and western boundary that transport different water masses from the north to the south and vice versa. There has been performed several monitoring activity of the basin through the presence of fixed observation points, which are part of the ICOS network. On the North Adriatic, we have the Miramare station, and in the Southern Adriatic, the EMSO E2M3A facility. These are two surface buoys composed by different sensors that measure either physical properties, but also biochemical ones. There are also moving platforms that are part of the biochemical Argo programs. So we have Argo floats, surface gliders, and in between 2019 and 2020, the basin was also monitored and explored by a surface sail drone. So we got also some information, high frequency observation from these platforms. And finally, we performed, thanks to the OGS, oceanographic surveys that in the station of Miramare, they happen on a monthly basis, while in the Southern Adriatic at the 2M3A facility, they happen on a yearly timescale. So the next slides will highlight some of the main features that we observed in the two basins. I will start with the main processes in the Northern Adriatic, and I will move then to the Southern Adriatic. So the carbon chemistry in the Northern Adriatic are mainly influenced by meteorological and hydrological variability due to the presence, to the effect of Bora wind, which is a northeasterly cold wind, and the influence of different rivers in the basin. This determines strong seasonal variability of physical and biochemical properties, changes in the stratification, and the production of dense water during winter, which influence the carbon chemistry. In the figure on the right, we see some time series taken from surface sensors deployed on the Miramare station that represents on the top temperature versus FCO2, and in the bottom salinity versus FCO2, where we see in the three different rectangles the events that I told you about before. So in the case one, there was a strong influence of riverine inputs that we observed because there was a strong drop in salinity, which increased the CO2 content at the surface. While in the other two situations, number 2 and 3, we had the effect of Bora wind that in one case happened in late spring and the other case in late summer. That we saw because of this decrease in temperature and increase in salinity, which contributes to increase the content of CO2. In the recent years, we have started also the monitoring of the carbon chemistry in the three main rivers, which are two actually that influence the Gulf of Trieste. I forgot to mention that our investigation for the Northern Adriatic focus in the Gulf of Trieste. So the two main rivers, which are the Isonzo and Timavo, and also the Aurisina, which is a spring of the Timavo River, we started to monitor and to take samples to understand the carbon chemistry of these rivers, and we observed that there were quite some differences. This was very interesting results, which is mainly due to the fact that the catchment areas of these rivers is diverse. In one case, for the Timavo, it's karstic, while the Zonso River flows outside the karstic area. So there is a different content of calcium carbonate, and also these two rivers, the Timavo River is an underground river, while the Zonso is overground. So there are different exchanges in CO2 with the atmosphere. And the presence of this local dynamics also allows us to get some insight on the buffer capacity of the Northern Adriatic when there is the occurrence of these different processes. The buffer capacity was calculated using the approach proposed by these two authors as expressed as the difference between total alkalinity and total carbon. When this difference is higher, it means that also the buffer capacity is higher, and this happens, for instance, when there is a strong river influence, while in the case of dense water formation, this buffer capacity decreases. Moving to the Southern Adriatic, we are exploring the effect of physical the changes in thermoline properties, and also the interaction of the different water masses that are present in the basin that influence the carbon chemistry. We have observed that in the latest year, either temperature and salinity has increased either at the surface and at intermediate layers. There was also a deepening of the vertical mixing, which has produced some effect on the carbon chemistry. Together with these processes, the presence and the interaction of the different water masses also produce some changes in the properties in the different layers of the water column, as highlighted by the figure on the right, that is a vertical profile, in this case of PCO2 in the winter months, where we see some vertical differences and increases in this case of the CO2 content in the different layers. We are also investigating surface dynamics thanks to the presence of a PCO2 surface sensor, and we are comparing our observations with model outputs to increase the temporal resolution of our data. And these investigations has highlighted that there is a strong seasonal variation of PCO2, which is strongly related to temperature. And also the use of model data allowed us to highlight this increasing trend, especially in the last 10 years in the winter CO2 content at the surface. We are investigating the surface layers because we are also interested to look at the CO2 fluxes in the region. Again, here we use the model observation, which are the violet and the light blue curve that we are comparing with our in situ observations, which are characterized by this the red curve. And again, the data highlighted that there is a strong seasonal signal. So the basin acts as a CO2 sink in winter and the CO2 source in summer. But also the model data highlighted that there is a strong variability, and there are some changes in the intensity of the fluxes during the winter period. So this data highlight an increasing trend of this of the intensity of the fluxes, and this is also a very interesting results that we have to deepen. And to conclude, what we would like to stress out with this presentation is that there are different processes influencing the carbon system in the two basins on different time scales. So for the Northern Adriatic, the two main processes are the influence of the different rivers, but also the meteorological forcing. While in the Southern Adriatic, the interaction of dense water, of different water masses and the production of dense water influence the carbonate system. On a general general base, the two sites act as a sink during winter because there is a strong effect of the winter physical pump, while in summer both regions release CO2. And we also would like to stress the fact that it's very important to pursue and to continue this integrated oceanographic approach, mixing data coming from different platforms, so fixed
Thank
points.
you so much.
Yeah, and
Thank you so much,
future
Carlotta.
okay, in future perspective, try to see which are the effect of these recent changes that also the Adriatic Sea is experiencing. And thank you.
This is very, very, very interesting. When will you defend your PhD?
I will finish in 2 years, so I'm just starting.
You are already almost ready, Carlotta. You are already almost ready to defend, I think. That's very, very interesting.
Well,
Thank you so much.
I still have to do a lot of things.
Yeah,
Thank
of
you.
course, of course. Thank you, Carlotta. And we are always actually happy to have early career scientists who are gonna present in the future sessions. You are more than welcome. The next speaker is my co-chair, Iris Hendriks. So Iris will actually present the greenhouse gases dynamics on the southern coast of the Mediterranean Sea. Iris, she's Dutch. However, she moved to Spain, where actually she was stuck there. And she didn't actually move. She's a very well-known researcher in the Mediterranean regarding many, many things. Actually, because she participated in over 25 national and international projects related to many aspects of ecology and biodiversity, from the study of marine microphyte habitats, in particular to seagrass meadows, their hydrodynamics, etc. But also lately, Iris, she was super interested in monitoring and in the time series monitoring in the Balearic Islands, especially regarding the measurements and monitoring of biogeochemical parameters and carbonate system measurements in that very interesting area as well. And she is also studying possible facilitation of calcification for associated fauna under future scenarios of ocean acidification. Iris is the co-chair also of the Ocean Acidification Mediterranean Hub, facilitating our exchanges and what is happening right now. So I would just give the floor to Iris. Welcome.
Thank you, Ahmed. Can you see my presentation okay, or is it full screen?
It's fantastic.
Perfect. So thank you. So I would like to present this work, even if it's very preliminary. Please keep that in mind. But it's a very interesting project that has been born from the MedHub community. I'm presenting this, but there's a lot of other authors who have done much more work than me on the project. So I think there's a lot of names missing here as well, because we had a lot of help during sampling. So before I start, a big thank you to everybody involved in this project. Yes. So how did this start? So this started by, because in 2018, we started a time series in the Balearic Seas, which we called BOAT, the Balearic Ocean Acidification Time Series. I have been talking about this time series before. We have been measuring pH, oxygen, salinity, temperature, and it's an opportunity to measure other parameters as well. So we were very interested in ocean acidification, but we were also measuring dissolved organic carbon, alkalinity, chlorophyll, to look at the drivers behind this ocean acidification signal and the trends that we were observing. And while we were just downloading data from the sensors, we were also measuring discrete samples monthly. We have 6 years of data at the moment. And every month we have been out there to download the data in the Bay of Palma and in Cabrera National Park, which is a very interesting station because it's very shallow, actually. So it's they're all very coastal stations, monitoring stations, not open ocean at all. So it's they have very complex drivers and complex processes behind. So while we were out there, we also measured methane and nitrous oxide in the water column. So this is all surface water, and it's all very close to the coast. So like you can see on this pictures here, And we were very interested in the drivers behind that. So while we were sampling, why were we sampling other greenhouse gases than the carbon dioxide? Because nitrous oxide and methane are very important drivers as well for the greenhouse gas effect. For instance, methane is more powerful than the carbon dioxide. And the potential of nitrous oxide is 300 times larger than carbon dioxide. So even if it's the most important greenhouse gas, carbon dioxide, it's also important to look at the other greenhouse gases. So that's why we also wanted to take them into account and measure them and see if we could find some trends as well in these signals. So if you look at the variability of the air-sea transfer of carbon dioxide, we see that it's not homogeneous at all. There's a big variability worldwide, but also in the Mediterranean, as we just saw as well in the last presentation. So the retention of carbon dioxide is not homogeneous at all. So we don't expect that for the other greenhouse gases either. And this is, for instance, a global map of the flux of methane, and you can see that it's also spatially very heterogeneous. But in the case of methane, it seems to be that the methane fluxes are much larger in the coastal area. So that is an opportunity for our time series, because we are actually monitoring the coastal areas. We are very close to the shore. So what we expected to see in our series is a higher signal of methane fluxes compared to the open ocean. So after a few years of data collection, we analyzed this data, and this is a published study. We published it in 2023. And what we found is that the Balearic Sea, so which is a very coastal area, it's also pretty anthropogenicized, that is actually a very weak source for methane fluxes. So that is very interesting when we look at the theory. So, and what about nitrous oxide? So nitrous oxide maybe is not such an interesting greenhouse gas to look at in our area, because it's mainly the highest fluxes are in the tropics, and most of it, 20%, is in coastal upwelling systems. So, and it has a very high seasonality. So what do we see in our time series? We see that indeed there is a pretty strong relationship between the saturation of nitrous oxide and the temperature. So what we expect is that in the Mediterranean Sea, which has a very different temperature in the surface waters, and also a very different trend in the surface area, with a very with much higher warming rates in the Eastern Mediterranean compared to the Western Mediterranean. So we expect that fluxes of nitrous oxide are also different between the Western and the Eastern Mediterranean, and that they will change differently in future scenarios as well, because the rates of warming are very different. So the problem here is that we don't have a lot of information about the southern coast of the Mediterranean, the southern and the Eastern Mediterranean. So most of the data is collected in the west and the northern parts of the Mediterranean. While these differences make us believe that just extrapolating the data that we collect in the north and the west will not do to calculate future scenarios of fluxes of the greenhouse gases in the Mediterranean. So that's why we asked for a project which is called Greenhouse Gas Dynamics on the Southern Coast of the Mediterranean. And we did this with some colleagues of the Mediterranean Hub to see if we could actually collect more data in undersampled areas. So the aim of this project was to evaluate greenhouse gas concentrations in the water column and eventually the ocean air fluxes. So I'm not going to present any fluxes because we haven't gotten around to that. We did a year, 13 months of sampling on the Moroccan coast, in Lebanon, and in Turkey with our colleagues. So we have this information, and we're still in the process of processing this data. That's why I say just keep in mind it's very preliminary. We just have put all the data together, and we're still processing it. But I really wanted to see you that this is to show you that this is the kind of initiative that can follow from the Mediterranean Hub, and there's scope to actually do more, because now we have collected data on the southern part of the Mediterranean, but as you can see, there's still big gaps, and I think there's scope to elaborate more on this project. So here in the middle is the Balearic Islands, where we have a lot of data available already, and the yellow dots are the places where we have collected more data. So to show you, this is very crude, very preliminary results, but what we see is that the methane concentrations in the surface water of the Mediterranean Sea averaged over the full year of sampling. We see that there are lower concentrations in the Western Mediterranean and higher when we go to the eastern part of the Mediterranean. And for nitrous oxide, it's actually the reverse. We see higher concentrations in the western part of the Mediterranean and lower in the eastern part. So these are interesting gradients, which we would expect, but there was no information available. So if we look at the trends over time, we can see that for methane, mostly what we see is that the variability increases at the end of our time series. Also, there are some outliers that can be explained by river plume, carbon input, but we have to, like I said, these are all the data. No data point has been left out. So now we have to see what caused these outliers and what processes are behind this. So what we see is that in the nitrous oxide, as expected, we see a very strong seasonal signal, which is very similar across the Mediterranean. And if we look at the relationship with temperature, we do indeed see the relationship that we expect with nitrous oxide and temperature. So if you remember, the other graph is going the other way around. There was saturation. So these are actually concentrations with temperature. So with a higher temperature, we see lower concentrations of nitrous oxide in the surface water. And also there's seems to be a relationship with salinity, mostly with nitrous oxide. So these are all very preliminary results. I just wanted to show you that we have this data, and now we're going to proceed to calculate ocean air fluxes and see if we can do an extrapolation of the fluxes on a basin scale. So hopefully I've shown you that this is very that we can collect very interesting data just on a larger basin scale. So it seems to be that there are east-west gradients, and we need to take them into account for a whole basin budget. So this was very short, so I hope that I have been able to make up a little bit for the time in the sessions.
You were fantastic. That was fantastic. Thank you so much, Iris. And yeah, I'm aware of this very nice project. Thank you. And yeah, we can go to the next speaker. Our next speaker is Nina Bednarsek. So she's affiliated to two institutes. So the institute the institute Jozef Stefan. I'm not sure that I'm pronouncing it very well, in Ljubljana, Slovenia, but also to Oregon State University in the US. So Nina, she's our passionate and experienced marine colleague with over 15 years of expertise in climate change mitigation, also on studying ocean acidification effects on many multiple marine biological groups, and also on marine carbon dioxide removal as well. And she also has been coordinating large-scale international projects and collaborating also with the stakeholders as well in order to find innovative climate solutions, but also to promote ocean resilience and carbon sequestration while ensuring the sustainability of our marine biodiversity. So, Nina, the floor is yours.
Thank you so much, and thanks for the invitation to this talk. It's always a pleasure to present something on a local scale as well. All right, can you see my screen?
Not in a presenting mode, yes, but not in a presenting mode.
All right, let's try again. This is okay, right?
Perfect.
Yeah, great. All right, so this talk is actually a combination of two different projects together. One is actually a NOAA study on ocean acidification enhancement synthesis, and the other one is from our basically conducted study within the OA Med Hub. So I put this together. So all the acknowledgements for multiple authors who have contributed to these studies are at the end, and I thank them all in advance. All right, so what have we learned from ocean acidification research in the past years, or decades, I should say now, is that basically negative biological impacts are already observable, specifically on the various marine calcifiers. But something that we have learned is basically how to develop various indicators and how to apply them to different systems as part of the monitoring strategies. And we have also extracted a lot of thresholds. They helped us in monitoring and model outputs in order to predict when we reach certain critical thresholds. So there is obviously an urgent need to mitigate against ocean acidification in order to manage and preserve our vulnerable species and habitats. And specifically here, I'll be talking about ocean alkalinity enhancement as a potential solution. This specific strategy is very much a potential strategy in the Mediterranean as well. And I always, and we as a community should always think about it to be useful or to be potentially implemented only if it doesn't cause any biological harm or risks. So when we are talking about the risk assessment, this is the IPCC scheme of how to think about that. And risks usually are combined of exposure, so something that can be part of the natural variability, but also part of the trends due to the climate change. Then obviously the sensitivity, a critical component of all this, where we try to streamline this process by focusing on the most sensitive species or life stages, and always try to connect the organismal or individual process to the population level process. Adaptive capacity is something that can alleviate sensitivity to a certain extent, so should always be part of the risks as well. And that usually includes the plasticity or genetic variability and also the spatial connectivity as well. This is exactly the same risk assessment that we have even a decade ago presented for OA, and you could see that when we were talking about the mCDR space, we could use exactly the same scheme. So thinking about the ocean alkalinity enhancement, there are exactly the same sort of strategies how the stressful effects could take place, either due to direct effects, and we are usually thinking about the individual organisms. Applying the stress means that we'll see some sort of genetic physiological changes, and they will be resulting in then physiological responses, behavioral responses, and in time potentially in the population level responses as well. Much more intricate are the indirect effects. And here we are thinking about the food web systems or ecological structures. And while we still have some direct effects, there are a lot of indirect effects, which are either known unknowns or unknown unknowns. And this is where potential really ecological ecosystem effects could come in. Specifically for the ocean alkalinity enhancement, we are potentially afraid that there could be a decline in primary productivity, failure of trophic levels, or in general loss of biodiversity. So all these things are something that we have to really understand. Sometimes experimentally we cannot address all these questions. So field work components or potential meta-analysis synthesis work is something where we are looking at as well. So when we are thinking about these mCDRs and thinking about building risk assessment, that's easy when you have a lot of data, but it's not easy when the biological implications and the studies have only just started. So how do we think about that and make the progress on something so important as the mCDRs? While we don't have that, how do we develop the monitorings and regulations in order to assure safety limits? And so here with a group of colleagues that, we were funded under this NOAA grant, we were thinking, how can decades of OA research and data available and archived in all these different archives can really help us with that? Because OA research has generated a lot of data. We have good predictive patterns. We understand so many different stuff. And so looking at the lowest hanging fruit, and those would be marine calcifiers, we now know that they exhibit different responses to ocean acidification. So we were thinking that if we really did a comprehensive literature review, analyze the data, and do some advanced statistical analysis, we could try to understand what would be happening across different species, their organismal responses, and try to determine some thresholds. And so I'll be presenting the results of this study. So we've actually covered 68 different studies, which encompassed 84 different species across 11 functional groups. Mostly the data was from the coral reefs, coralline algae, and the mollusks, and then different other functional groups represented in this diagram in here. So we were looking specifically on the data on the classification rates. And then the idea was to conceptually adding sodium hydroxide to be able to statistically predict the responses under high pH, which is related to the concept of ocean alkalinity enhancement. So these are the categories of different responses that we basically gathered from ocean acidification studies. So organisms can either respond in linear positive, negative response, threshold, positive, negative, or so-called exponential responses, parabolic, which are physiologically actually the most sensitive responses, or neutral response. So there is no response. And what we have done then upon that is adding a base, so either sodium hydroxide or sodium carbonate in different increments. And so then we calculated corresponding classification rates using the regression models. So this, and then you can see still where are potential thresholds happening, where we can see any sort of shift in the responses and so on. So this is now an accepted study in Hydrosphere, and we have also published all the codes. So if you want to check how your local species maybe for which you have OA data available, how they might be responding to ocean alkalinity enhancement, you can do that with the help of all the open source tools that we have put in this study. Okay, so what we have found out is that basically we'll have, across all these species in different functional groups, we would have approximately 40% species that will have neutral response, which is perfect. This is what we want. We don't want ocean alkalinity enhancement to introduce any positive or negative response. We'll have 25% negative. So imagine 1/4 of the species would actually be negative responders. So their classification rates would decrease, and the most sensitive species would be echinoderms, coralline algae, dinoflagellates, and crustaceans. Some of them would also be positive responses. In the grand scheme of ocean acidification, those positive responses could be actually good, but we are very careful not to, we should be careful not to induce these potential shifts in the communities when we start having the winners and the losers in the system, and that might introduce further implications, biogeochemical and ecosystems. So this sort of study, I think, is very useful, not because it's very accurate. These are all the predictions and they need to be tested, but because it really provides the guidance on the most sensitive species and the species where we have no concern, the reason for concern. And why that's important is because in the next maybe 5 to 10 years time, in order to catch the industry, which is really fast in trying to, come to the point of implementing OAE for carbon credits, the science has to produce basically a basic knowledge how these things will ecologically impact the community. So if we can actually save time and say, okay, we have to test these species because we know they are the most sensitive versus the others that, we know that are not among the most sensitive, I think this is a major breakthrough that we could actually achieve with a synthesis study like this. Okay, thinking about, testing in the field results, it's really important that we start experimenting with much more realistic conditions that are specifically in the field. And experimentally, right now, even the biological studies are using much higher dosages that would ever appear in the field. And I think if we start implementing lower exposure and more realistic conditions, that also might mean lower exposures and lower risks. So certain risk could still happen, but I think we can understand which species and processes we need to monitor in order to potentially catch these negative implications. So importance of field trials is there. And today we have already heard from Carlotta about this Mediterranean being, this very dynamic region with very strong west-east divide in total alkalinity gradients and other carbonate chemistry parameters as well, with high total alkalinity gradients in the east. And the idea was that we could actually do some biological and biogeochemical studies and try out the implications in there. So stuff that is potentially impossible to do in the experiments, we could actually do in the field. And so thinking about the potential OAE natural analog multiple site system. So something that is, that Cristina Gambi presented in the southern Italy, we could actually have across the entire Mediterranean. And you could see from IRIS how successful different coordinations are in the MET. And so we are also thinking how OA Met Hub can act as a facilitator of the network to form this working group on the natural analogs. Prior to this, we wanted to actually test a little bit how different people and various stakeholders are thinking about the mCDR in the MET space. So this summer, we have created the questionnaire with 40 questions and then distributed this questionnaire to 50 different stakeholders. And I'm just briefly going to present some of these results in here. So we had responders, and to our surprise, there were 50% approximately of all people who were actually governmental research and 40% in academia. So in this particular case, our primary stakeholders were actually experts in the government space. And they were equally distributed between East and West, which is perfect for something like this. And they all, most of them were not involved in mCDRs, but maybe some others were already involved in some adaptation strategies going on in the mCDR world. But most of them really express an interest in collaborating or expanding in the mCDR-related work in the MED. So then when we ask what sort of aspect of mCDR would they consider, and they obviously realize that biological and biogeochemical responses, so studies are very important, but here you could see the studying economic aspects and policy, diplomacy, and governance was actually also specifically emphasized here. And this, I think, stems from the fact that people really think about potential obstacles for the mCDR in the MED, and probably that's not just the case in the MED, but wider scales and probably global as well. And where are the potential obstacles? They are not in the natural systems, that they are in the political, administrative, legislative, and economic systems. So those are main obstacles that were recognized. And I think this is probably one of the primary results that we want to think about, and how to basically start the research knowing where the potential obstacles are. And here also the same sort of results that reflect these obstacles. So where do we have to work to further advance the topic of mCDR? Obviously, the policy and management got the higher score, and then somehow equal distribution between the research and actually the legislation that could enhance the mCDR developing in the region. I think that's very telling. Obviously, the community is very much involved in thinking and how to go about that, and trying to actually come to the consensus of how to do that. Because when we ask, could the Mediterranean be that sub-basin where the network of OAE natural analogs could be developed, they all pretty much in consensus agree that that could be the case. But also pay attention to this, right? A lot of actually speakers realize that the current scheme of the networks is not sufficient. It's lacking mCDR expertise, and specifically the new biological nodes need to be established, right? And so they recognize that this could be somehow additional networks of coastal marine stations that could specialize in OAE and then the scientist would not be, working individually on the problem, but those could be specific biological nodes where the biogeochemical and biological processes could be integrated. So I think that's very telling in what's going on in the med space, and this is where we as a OA Med Hub want to take this further. So stay tuned, and we'll see what we have in approximately a couple of next months when we start analyzing the results of this study. So for now, thank you for this, but I really want to acknowledge all my authors who have contributed to either the first part of the study, the synthesis work of, or the OA Med Hub that has only just started with this questionnaire, and the survey will be coming in with the results later on in the year.
Thank you so much, Nina. Thank you. This is a very promising effort that is proudly led by us, by the Med Hub, and we thank Nina for the leadership, and hopefully you will be able to see the product of this very, very well soon. So let's jump to the next and the final, actually, speaker, Valeria Ivello, our colleague. Valeria, actually, she's now affiliated to the CNR, so the Institute of Marine Sciences, that is affiliated to the National Research Council in Italy, ISMAR. But actually, Valeria, she is a biogeochemist, and she was a lecturer at the Middle East Technical University Institute of Marine Sciences, METU, from 2013. She was there teaching about marine biogeochemistry, ocean acidification, global biogeochemical cycles, and she has a long expertise also in conducting research in the Mediterranean Sea as well, especially in the Eastern Mediterranean Sea, related to the monitoring actually of the CO2 fluxes and dynamics, air-sea interfaces, etc. And yeah, she has very also good knowledge about nutrients, dissolved organic carbon, especially in poor marine environments. Valeria, she's also leading one of our hub's efforts, the intercomparison exercise, that we are so happy that she's leading it, and she's gonna present also the preliminary results. The floor is yours, Valeria.
Hi, Ahmed. Hi, everybody. Thanks a lot for having me here. And I'm very glad to finally present this effort that started already 1.5 years ago. I hope you can see my screen now.
Not in a presenting mode.
Okay. What about now?
Perfect, yeah.
Okay, so yeah, first a little bit information about the context of this intercomparison exercise. 1.5 years ago, we were together in Turkey for a steering committee, and we wanted to use this opportunity to be all together to make profit of, to actually to realize one of the ideas that we were talking since a while. So to do an intercomparison exercise of marine carbonate system parameters in the Mediterranean Sea. So when we were there, basically a couple of days before, so you can see here all the people that took part to the experiments. Some of them are from the steering committee, some others have joined, they are still part of Mediterranean Hub, but have joined the experiment thanks to the colleagues. So you see here we have a very big number of authors, co-authors, and we have 15 laboratories here. So when we were there a couple of days before, we decided to collect, I mean, of course, we planned much in advance, but we collect all the samples. We went to this site that is in the southeastern Mediterranean Sea. This is the Turkish coast, the southern Turkish coast. This is Cyprus. So our sampling station was offshore Turkish coast, 500 meters depth, and we collect samples from 250 meters. The idea here was to, of course, to avoid to avoid as much as possible biological activity, and to have a proper like a plain sample. So we follow some specific procedures to collect the samples. We collect more than 500 liters. We mixed in a very big tank for a couple of hours, and then we left resting for 24 hours. And then the following day, after a gentle paddling, we sampled for a duration of 3 hours. All the bottles were carefully prepared, so they were first acid soaked, then it was washed. They were washed with still water, and also put it in a muffles. So the intercomparison parameter we decide to compare is a pH, total alkalinity, and DIC. So every lab received 5 replicates, 5 bottles. Some lab received 2 times because they distributed to other labs. In total, we collect 80 samples. And in order to check the variability over time of the samples, the Mediterranean, the METU, so the Middle East Technical University, so the institute I was belonging to, took it 3 samples, three batches. Okay, so in order to have it a sampling soon after the sampling, a sampling after 2.5 months, and a sampling after 6 months. And this is what we did. So these are the laboratories, the list of laboratories you can visualize better on the map of the Mediterranean and Europe. So in total, there are 9 countries involved, 12 laboratories, and the timeline of the analysis is 244 days. So from the very first till the last lab. So as Abed mentioned, this is a preliminary study. So I will show you some first results. Our focus at this stage is to first to understand if there were any biases on the sampling that were collected in terms of the beginning of or the end. So if there was any effect of these 3 hours that were used for collecting the samples because of temperature, because of exchange of gases or whatever. And also another question we had in our mind, if there was a time lapse, if there was an effect of time lapse between the different time where the analysis were carried out from the different laboratories. And finally, the most important, if there's a difference between the different laboratories. So the first parameters, this is the dissolved oxygen, the results of dissolved oxygen collected during the sampling, the collection of the samples. Okay, so every, you see here 12, because every 6 samples, we collect an oxygen sample in order to have a check during all the sampling, during all the 3 hours. So you can see here brief statistics. There's an average and standard deviation and the coefficient of variation. So basically, we can say that there was absolutely no impact, no variation in terms of oxygen changes during the sampling. So this is the first results of pH. So we show, as I said, that we show pH, total alkalinity, and dissolved inorganic carbon. So this is the pH measurements. We assigned the code to all the 12 laboratories that took part to keep anonymity. And you can see here, there's a variability over these 12 laboratories. Here you have a brief statistics. So in total, the number of pH samples that were analyzed in terms of bottles, then every bottle received different replicates. It was 39, and then the total number of replicates that were carried out was 120. Labs that did the things, this was 8. And the time that passed from the first analysis till the last one was 240 days, so about 8 months. And the average we have is 7.95. And you can see also the standard deviation is not pretty bad, it's 0.09 with a minimum maximum varying of 0.04. Now, when we look at the sample sequence, so the question here is do we have any trend? For example, at the beginning it was very good, and then at the end there was the end of the sampling, there was some problem. Well, actually we applied these statistics, and apparently, as you can see from this R2, there's no linear trend that is associated to this. So basically we can say that all the sample, as we saw from the dissolved oxygen, all the sample didn't really show any variation over time. When we check on the other side the effect of different time for the analysis, well, here the story is a little bit different. So this is at the very beginning when the analysis have been done. Most of the lab actually have carried out this analysis within the first 40 days. This is the second step where the METU has carried out the second analysis, and unfortunately for some issues, these samples have been done, but I'm not presenting here the results. And then we had the other two labs, and this is again METU. Here I need to show because we are the reference. I cannot keep the anonymity because we were the references to show the variability over time. But what is interesting here to observe is that when you compare this first data set and this last data set, we found that there was a significant difference, meaning that probably the effect of so much time storage had an impact on the sample analysis. And this is also reflected by this other analysis that was carried out just like 10, 15 days before the last analysis that also show lower values. So we came to the conclusion that probably, at least for pH, there was an effect of this long storage of 8 months. When we remove these two samples and we carry out the same statistics, we see that when we look at the standard deviation, we have a lower values, even though, I mean, there's a minor difference between the two samples. The coefficient of variation, of course, improves. When we look now on total alkalinity here, the number of labs that could realize these measurements were higher. So we had 9 labs. Overall, you can see there was a general average, a general agreement between all the labs. A part of this funny things that happen on the first lab that need to be investigated a little bit further. So this lab had, like the others, they had 5 samples, and apparently 2 samples gave a very high and low values. This need to be understood. I believe there was some technical and some technical issues, because the other 3 samples they had were perfectly in line with all the other analysis. So this should be some issue that need to be checked. So when we check again if there's a trend or any effect of time sample sequences, again, you can see that R2 is extremely low, so we can assume there was no effect on this. And when we check we check pardon, this is not sample sequence. So yeah, this is the time lapse of this. Of course, when we have this too big variation of 2 samples that actually change a little bit the scales, and so you cannot really assess. When we remove these 2 samples and we check, we can see there is a sort of trend. So total alkalinity, while we saw that pH was decreasing, we see here that somehow total alkalinity seems to improve, to change actually to the very first. And this is what the statistics show. So we observed there was no statistic difference between April and July, but when we compare this December analysis with April and July, we found that there was a significant difference. So probably, again, there was an effect on of storage on the total alkalinity. But this is, I would say, this is still preliminary. So I would check again more carefully this data. This need a little bit further investigation. When we look at the DIC actually was carried out by a lower number of samples, because not every lab has the instruments to measure this parameter. So you can see here that there's a good alignment of a good convergence of this lab, a part of lab number 5. And again, we don't really know which is the reason because of this, and we need a further investigation. So coming to the conclusion, because time is finishing, so there's no special trend among the sample associated to the sampling sequences. We found that there's an intercomparison of pH among the 8 laboratories, show a limited variability. Possible effect of long-term storage has been observed, and the same observation is valid also for total alkalinity that has been carried out in the 9 laboratories. But there was this lab, the lab number 1, that showed these 2 extreme high and extreme low values that need to be more investigated. So also here we found there was a possible effect on long-term storage. Again, this is a need to be experienced better. Similar aspects have been observed for the IC, and we have 4 laboratories, and the 1 lab out of these 4 was a little bit out of range, so this need to be understood the reason. So next step for this study, I mean, again, I say this is a preliminary analysis. So we need to go further with the statistical analysis of the 3 parameters to understand if there's actually a significant difference between the all the different analysis. But I mean, seems already quite in good agreement. So we need to assess which is the impact of the dye. We also, I didn't show here in detail, but some labs carry out the analysis of pH, spectrophotometric pH with the purified and purified. So we need to make an analysis on this to see if there's a clear signal resulting from this. Also, a detailed analysis on lab procedures. So temperature condition, internal lab use, and reference materials need to be analyzed. And also, sorry, there is some typo here, identify some, I mean, at the end of the study, our purpose is to identify some key recommendation for carbonate system analysis in the Mediterranean Sea.
Fantastic. Thank you so much, our colleague Valeria. Thank you also for leading this very nice effort of us. That is also, thanks to IMEDEA for sure, and for your leadership in this. And stay tuned for the results. Hopefully very, very, very soon. Thank you, Valeria. I will give the floor now to Iris. So Iris, please, you can check if there are questions. Maybe we have a couple of minutes for that.
Yes, so we only have answered questions at the moment. So maybe some of the people that asked for questions want to elaborate, want to ask something more. For instance, Michele, I saw that you have asked about the metals in the vents.
Yeah.
Or maybe Anna Franco, who also had a question, maybe wants to elaborate on that. Also, if there are any new questions, please put them in the question and answer box.
These people are shy, but you have to bear in mind that you have to bear in mind that our session is recorded and it will be uploaded on GOA-ON's YouTube channel. So also, if you have any questions, you can also put them there. And I mean, we can check the questions there, but also you have now the contacts of our speakers. So if you have any questions, you can directly contact them as well. We would like to thank you all for your presence, for your attendance. We would like to thank our speakers. the 5 scientists. Thank you so much for dedicating your time, and best of luck in your studies and efforts, initiatives that you are conducting. And yes, so that was it, or do we have something else? So Michele, yeah, he would like to speak, not to write. You can speak, Michele.
Maybe that's not possible for the attender, the people that attend. Is that possible?
I think it, but well, I mean, well, we all, it is possible based on Caterina, but it seems that maybe there are technical issues. So we would like to close the session for now. Thank you so much, everyone.
Just joined.
Yes.
Okay, yeah.
Can you hear me?
Yes.
Okay. No, yes. No, very interesting presentation, Iris, but yes, I was wondering, yes, on the effect no, of temperature, if is more driven, yes, by physical or biological factors.
Yes, that's
or NO2 concentration, yes.
Yes, that's a very good question. And since I just saw the results this morning, would require a little bit more thought. I would at first thought I would say physical, but we have data on biological drivers like chlorophyll and other parameters, so that would need some exploring. It's a very interesting question, though. I hope to be able to answer it very soon.
Okay, thank you very much. I will also be very curious about this. Yes, and for Maria Cristina, yes, she already answered to my questions. I'm interested in what are the possible effects on trace metals of this lowering of pH in these areas, because I think that the change of pH is very complex, is affecting many equilibria, and so also the complexation of metals that can exert it may be a toxic effect of some organism or not, and so is a point very interesting to better understand, I think, in these areas.
Yeah, Michele, I agree with you. That's the metal is still and in most of the events is still an open question. But in Vulcano, they have measured, and there is an excess of ions, especially of iron. And this seems that is the cause that why differently from other vents, the Cimodocea in the Vulcano vents is not so flourishing. Is more dense. It thrives better outside of the vent than in the vent. It seems that this excess of iron, especially iron, and if you look at the underwater, the Vulcano vents, there is this red film all over the rocks because of this excess of iron. So this is a problem that has been highlighted by people studying the vent there, especially the paper of Vizzini and al., where they highlight this potential problem, this confounding effect. But in Ischia, fortunately, we had some measurements in the water, and there is no such excess of metal ions respect to the control station. So every event is different.
Thank you both, Maria and Michele, for your questions. I think there are no additional questions for the moment. We have to close the session. Thank you all. And it's going to be uploaded on YouTube. So thank you all, and till next time, hopefully. Bye.
Goodbye.
Bye-bye. Thank you.
Bye-bye.
Bye.